Welcome to Aquatic Biology. Today, we're going to explore a fascinating but highly destructive process known as eutrophication. If you have ever seen a lake covered in a thick layer of green slime and wondered what caused it, you are looking at eutrophication in action. This topic is absolutely essential for understanding how human activities impact our freshwater ecosystems. And by the end of this lesson, you will know exactly how to measure and classify the health of a body of water. Let us start with a simple analogy to build our intuition. Imagine you have a beautiful, clear garden pond. One day, you decide to dump a massive truckload of plant fertilizer right into the water. You might think this would make the pond healthier, but instead, the microscopic algae in the water go absolutely crazy. They multiply so fast that the water soon looks like thick green pea soup. Because this dense green layer blocks the sunlight, the plants at the bottom die. As they rot, they suck all the breathable air out of the water, leaving the fish gasping for breath. This tragic chain of events is exactly what happens in the real world. Now that we understand the basic idea, let us look at the formal scientific definition. In the study of aquatic biology, this destructive process is known as eutrophication. The word itself comes from Greek, where eu means well or good, and trophy means nourishment. So it literally translates to well-nourished or over-enriched. Eutrophication is defined as the process by which a body of water becomes overly enriched with dissolved nutrients. Specifically, we are talking about nitrogen and phosphorus. In an aquatic ecosystem, these elements act as powerful fertilizers, triggering the exact same explosive plant growth we just imagined in our garden pond. Understanding the definition leads us to the actual biological mechanism. When that unnatural spike in nutrients hits the water, it triggers an explosive, uncontrolled growth of algae, which we call an algal bloom. But the algae do not live forever. When these massive blooms eventually die, they sink to the bottom of the lake. Here is where the real damage happens. The bacteria that decompose this dead algae consume vast amounts of dissolved oxygen from the water. This rapid oxygen depletion leads to a deadly state called hypoxia. In a hypoxic environment, aquatic animals simply cannot survive, leading to massive die-offs. So why is this chain reaction so important for us to study? Understanding eutrophication is absolutely critical because it is one of the most widespread and severe water pollution problems facing our planet today. When a lake or coastal area becomes eutrophic, it loses its ability to support diverse life. It literally turns vibrant, thriving ecosystems into barren, dead zones where no fish or shellfish can survive. This ecological collapse has devastating real-world consequences for human communities that rely on these waters for their livelihoods, particularly those dependent on fishing and recreational tourism. Beyond the economic impact and a loss of fish, there is a direct threat to health. Some of the algae that bloom during this process are known as cyanobacteria. These specific bacteria produce dangerous toxins that contaminate the water supply. These toxins are highly poisonous and can severely harm pets, wildlife, and even humans who come into contact with or accidentally ingest the water. If we did not study and monitor this process, we would have no way to regulate agricultural runoff or treat sewage effectively to protect our precious freshwater resources from these toxic blooms. To manage this pollution, we need a way to measure it, which brings us to our first mathematical tool. Scientists use Carlson's trophic state index to assign a numerical value to water quality. Since phosphorus is usually the primary nutrient triggering owl growth, we calculate the index using total phosphorus. The formula is TSI equals 14.42 times the natural log of TP plus 4.15. TSI is a dimensionless number, usually ranging from 0 to 100. TP is the concentration of total phosphorus in micrograms per liter. The natural logarithm helps compress a very wide range of phosphorus measurements into a neat scale. As phosphorus increases, the index score increases, indicating worse pollution. Now that we have our formula, let us see this with a concrete example. Suppose an environmental scientist collects a water sample near heavy agricultural farming and finds the total phosphorus is 65 micrograms per liter. We need to calculate the trophic state index. First, we substitute 65 into our equation in place of TP. Next, we evaluate the natural logarithm of 65, which gives us approximately 4.174. We then multiply this by our scaling factor of 14.42, yielding 60.19. Finally, we add the constant 4.15. Our final answer is a TSI of 64.34. Because this score is well above 50, we classify this lake as highly eutrophic and severely polluted. Having seen how chemical measurements work, let us look at a physical measurement technique. Another common way scientists measure eutrophication in the field is by checking water clarity. They use a simple tool called a Secchi disc, which is a heavy black and white painted disc. The scientist lowers this disc into the water on a rope until it completely disappears from sight. Because algal blooms make the water murky, a shallower disappearance depth means there is more algae and therefore more pollution. This physical clarity measurement gives us another way to calculate the health of the lake. This physical measurement leads us to a slightly different formula for the trophic state index. The formula based on Secchi depth is TSI equals 60 minus 14.41 times the natural log of SD. Here, SD represents the Secchi depth measured strictly in meters. Notice the negative sign placed right before the logarithm function. This is crucial. It mathematically captures the inverse relationship between water clarity and pollution. As nutrient levels go up and algae multiply, the water clarity naturally goes down. The negative sign ensures that a smaller depth, meaning murkier water, correctly results in a higher, worse index score. Let us apply this new formula to a field measurement. A lake manager lowers a Secchi disc and finds it disappears at a depth of 1.5 meters. We want to calculate the trophic state index. First, we substitute 1.5 directly into the equation for SD, ensuring it is in meters. Next, we evaluate the natural logarithm of 1.5, which is approximately 0.405. We multiply this logarithmic value by 14.41, giving us exactly 5.84. Finally, we subtract this from our baseline score of 60. The final answer is 54.16. Since this is above 50, the lake is officially eutrophic, clouded by a heavy suspension of microscopic algae. Now that we have calculated the severity of the pollution, let us visualize exactly what is happening beneath the surface. On the left side of this diagram, you see a healthy, oligotrophic lake. Notice how the sunlight penetrates deep into the clear water, allowing aquatic plants to thrive at the bottom and providing plenty of oxygen for the fish. On the right side, we see the eutrophic lake. Nutrient runoff from the nearby farm has fueled a massive algal bloom at the surface. You can clearly see how this green layer blocks the sunlight. Without light, the bottom plants die and decay, which depletes the oxygen, leading directly to the fish kills shown at the bottom. This diagram perfectly summarizes the tragic biological shift we have been discussing. We have covered a lot of ground today, so let us review our key takeaways. First, eutrophication is a destructive biological process triggered when aquatic ecosystems receive an unnatural overload of nutrients, specifically nitrogen and phosphorus. Second, this excess food causes algae to multiply uncontrollably, blocking sunlight and ultimately depleting the water of life-sustaining oxygen. Finally, we learned that scientists do not just guess how polluted a lake is. They use mathematical tools like the trophic state index. By measuring total phosphorus or physical water clarity, we can accurately classify the severity of the pollution. Understanding these concepts is your first major step in learning how to protect and conserve our vital aquatic biomes for future generations.